Hello everyone, I'm Mr. Furlong, and today we're going to be talking about the process of photosynthesis. So here I am today out in the middle of a woods filled with mosquitoes, helping you learn this process of photosynthesis. This is an amazing process that's going on. Without it, there'd be no animals. We could be living on a planet of just plants. Certainly they don't need us, but we need them because they are the basis of the food chain. They are what is providing food and energy for us. And so let's take a little bit of time and go through this process. Starting with the light reactions. Now remember that the light reactions occur on the thylakoid membrane. So here you can see the lipid bilayer, and embedded in it are proteins. Now some of these proteins are going to be acting as enzymes, some of them are going to be acting as transport proteins, and yet others are going to have chlorophyll located in them to absorb the energy. As we go through this process, you can jot down in your notes what's happening at each of the different stages. Notice that in this diagram there are certain letters that are shown. For instance, we have letter A. That's going to in essence be kind of the first step where we're going to be going to. Letter B, letter C, and so on. First of all, light energy is being absorbed, and it's being absorbed in two specific locations. We have, notice here it's PS2 and PS1. Now that doesn't stand for PlayStation, that stands for Photosystem 1 and Photosystem 2. This is what contains the chlorophyll, and it is absorbing the light energy, and it's causing electrons to get excited and move to higher energy levels, but we don't need to worry about that. Right. What we want to do is look at how this energy is going to be used and follow this flow of energy throughout this process. This light energy is going to be focused to a small section of the photosystem known as the reaction center. In the reaction center, this is where the water is going to be split into oxygen, which is then going to be released, and hydrogen. Remember, hydrogen is simply one electron and one proton. The electron is going to start to travel through this electron transport system, whereas the hydrogen is going to be released inside of the thylakoid. Let's follow the flow of electrons, because really when you think about a flow of electrons, what comes to mind is electricity. There's energy there. And so these electrons are going to be moving along this electron transport system. And real quick, of course, these electrons are moving from atom to atom. They're literally jumping from one atom to the other atom. Now when this happens, of course, it's like this little electrical current. Now that is going to provide energy then for, here at step D, hydrogen that is on the outside part of the thylakoid to get pumped to the inside of the thylakoid. Now what's happening, of course, is that we're building up a whole bunch of hydrogen ions inside the thylakoid membrane. Now that's important because at step E, hydrogen is going to diffuse through this big protein here. It looks kind of like a light bulb, and it really does. Part of it's spinning around and so on, but as hydrogen goes from an area of high concentration to an area of low concentration, that's going to allow an ADP to add a phosphate to it and become ATP. Now remember, this ATP then is going to head on over to the Calvin cycle. At the other end of this diagram, of course, we're having all these electrons. Now these electrons need a place to go. So they're going to combine with these hydrogens and an NADP molecule to form NADPH, which again is carrying a little bit of energy along with ATP over to the Calvin cycle. Now remember, the Calvin cycle is taking place out here in the stroma, so it doesn't have to go far. In the Calvin cycle, we're going to start with carbon dioxide coming in, and there's actually 3 carbon dioxides that's going to be used along with 3 of these RuBPs or rubisco biphosphate, and it's going to form a 6-carbon molecule, and specifically it's going to form 3 of those 6 carbon molecules. This is known as carbon fixation. Now immediately an enzyme is going to come along and it's going to break that 6-carbon molecule into 2 PGA molecules. What we really have then are 6 PGA molecules. Now here's where the ATP and NADPH are going to come in. They're carrying a little bit of energy that ultimately they got from the sun, and they're giving it to those PGA molecules. Now this changes the molecule just slightly, and so now it's no longer PGA, but it's called PGAL. Here we are going to have one of these PGALs is going to actually become half a glucose molecule. The other 5 PGALs that are made are going to continue through the help of enzymes to be regenerated. And so we can see that one PGAL drops out of the cycle for every 6 that's made. And of course, remember, it takes 2 PGALs to make that one glucose. But anyways, these enzymes are going to regenerate those PGALs back into those 5-carbon RuBPs, and specifically there's going to regenerate into 3 RuBPs again, along with the help from another ATP molecule, and this process can now start all over again. And as long as there's light energy, as long as there's plenty of carbon dioxide, this process can continue basically until the sun goes down. Just a fascinating process. Took many scientists a long time to figure this out, and here you can learn this in just a short amount of time. Isn't technology great? So we are going to start on a great lab experiment over the next couple of days where we're going to manipulate variables to find out how they affect how fast photosynthesis takes place, and I can't wait to get started with you on that. See you then.